Hello everyone, my name is Yugo Lima-Melo. I'm a postdoc at the Federal University of Rio Grande do Sul, and I'm here to present a study by me and my colleagues Breno Gonçalves, Professor Felipe Marasquim, and Professor Márcia Magis Pinheiro to the Bioinformatics from Algorithm to Applications 2020 event. So the title of our presentation is Transcriptome Meta-Analysis of Low Phosphate-Treated Rice Roots Reveals Novel Players in Phosphate Starvation Response of Crops. So as you can see by the title, we are plant researchers, and although we do not perform deeper bioinformatic studies, we use bioinformatic tools trying to answer our scientific questions. So this is what our study is about actually in this event. So the thing we are trying to answer here or to solve here is the phosphorus problem. So the phosphorus is an essential element for life, and not only for plants but for animals and other kind of lives. And for plants, it's a major essential macronutrient, and being so, it is important for crop yield and food production. In parallel, phosphorus is scarce and has non-renewable availability in soil. So added to these two facts, population is growing and consequently demanding for more fertilizers like phosphorus in agriculture, for example. So there is a clear need of improvement of crops to achieve higher phosphorus efficiency. This means that we are looking for crops which can better use or had better uptake of phosphorus for food production, for example. So this is the general aim of our research group, one of them. And for this, we need first to understand how plants deal with low inorganic phosphate, which is the typical form of phosphorus in biological systems. So plants can sense. Actually, plant first has to sense the phosphate status. So this can occur locally in roots or systematically in the whole plant, roots and shoots. And when plants perceive that they are under low phosphate concentrations, they develop the so-called phosphate starvation response in an attempt to improve phosphate use efficiency and uptake. And it is totally dependent on the regulation of phosphate starvation genes. So plants can sense the phosphate status, as I said, with these two mechanisms, the local one and the systematic one, and each of them has different cascades of events and different activations, but in the end they develop to the same result, which is the activation of phosphate starvation responsive genes and the development of phosphate starvation response. And not going into details, but speaking very briefly, phosphate starvation responses induce morphological, physiological, biochemical, and molecular response. And some classical examples are the changes in the root system architecture and the root secretion of organic acids on phosphatase. And these are some examples that make the plants able to improve their phosphate acquisition. However, most of the events related to phosphate starvation are still poorly understood in crops when compared to Arabidopsis thaliana. This is expected because Arabidopsis thaliana is the model for plant science. However, we have to keep in mind that crops are ultimately important as well. So we need to understand these events also in crops, because crops produce food which is important for humans and animals in general, and it's important for the hungry issue we have in the world. So here we are focusing in rice, specifically Oryza sativa, which is a mother plant for monocots, crops, and cereals, and it is one of the major staple foods worldwide, and it has a high demand for phosphate fertilizers. So the mechanisms to face phosphate starvation needs to be accurate. So based on all this information, we very recently, 2 weeks ago, published this paper in the Environmental and Experimental Botany with the title being Phosphate Starvation Responses in Crop Roots from Well-Known Players to Novel Candidates. And here we discussed the recent progress on remaining gaps of knowledge on the molecular mechanisms of sensing and signaling phosphate starvation in plants. And we also explored publicly available transcriptomic data of rice roots using bioinformatic tools. And this is the focus for this presentation. We're not going to the first topic, but actually to the second, which involves more bioinformatic things. So with this, we were able to propose new functions on phosphate metabolism for previously known genes, and to identify attention-worth genes with unknown function in phosphate metabolism. These were our aims during the development of this study. So for this, we analyzed transcriptomic data available in the Gene Expression Omnibus Public Repository, the GEO. And there we were able to select all data sets available unambiguously specifying the comparison between transcriptomics of rice roots after phosphate starvation treatment and rice roots grown under a phosphate sufficient condition during the same period. So we had the control and the phosphate starvation treatment for all data sets selected. It's important to highlight that we only used Oryza sativa, cultivar nippon because we wanted to have plants with not the same genome, but I mean genetically similar among them, so we could compare the different data sets, which would not occur with if we had, for example, different cultivars. Some of them are phosphate tolerant, phosphate starvation tolerant, so others are phosphate starvation sensitive. So we have to focus in only one. And after that, we selected only significantly regulated genes based on the data and the information of each experiment for each data set. So a total of 7 data sets from 4 independent experiments matching these criteria were found and used. You can see here in this table the description and some information for each data set, like the data set ID, the geo series, where they can be found, the name of the samples, and also the age of the plants and the period of phosphate starvation in each experiment. And as you can see, they are different among the data sets, but it is expected because researchers are always using different kinds of treatments and plant ages, so it was expected. However, even with this difference, we were able to find genes which had the same kind of response in all data sets, or at least in most of them. For example, considering all the 7 data sets, we found 8 upregulated genes and 3 downregulated genes. Also, 33 genes were upregulated and 37 genes were downregulated, considering at least 6 data sets. And we made this our working group, because we had the feeling that 6 data sets is a good number to give us some confidence on the methods and on the analysis for the analysis. And at the same time, we had a good amount of genes to investigate. And considering the total of genes which were regulated, we had 12,000 upregulated genes and 13,000 downregulated genes, which means that these genes were regulated in at least one of the data sets. So we were able to pinpoint the genes which were downregulated or upregulated in at least 6 of those data sets. And as you can see, they displayed different levels of expression among the data sets, but they were generally consistent among the same data sets. So, for example, we didn't have any case of gene that was upregulated in one data set but downregulated in the other data set. So this also gave us some confidence on the shoes of the genes to explore. And here you can see the general clustered heat map showing the expression profiles of the down and upregulated genes. So after finding these genes, which are all of them are phosphate starvation responsive genes because of the treatment of phosphate starvation, obviously. So we performed a deep search on literature for information for each of these genes. And then we were able to classify them into groups. The group of genes with non-function in phosphate metabolism and the group with genes with unknown function in phosphate metabolism. So each of these groups had the same number, interestingly, which was 35 genes for each group. And the group with non-function, they can mean that they can mean common phosphate starvation response in rice, not only in rice but probably in other species. While the group of non-function means new targets for new findings. So here you can see the table for each one, and first focusing in the group of genes with non-function in phosphate metabolism. First, we found that several genes are Marquee genes for phosphate starvation. These also gave us confidence on the selection of the genes and the methods we use to reach them. But I would like to highlight here that some of them, although being genes with non-function in phosphate metabolism, they are also worth of deeper investigation in our opinion, because although they are listed as responsive to phosphate starvation and they have a role in this metabolism, these roles and these functions are not well established, because sometimes they are hypotheses or they were never specifically tested, experimentally tested with these genes, but sometimes with a close member of the gene family or something like this. So in our opinion, some investigation is still necessary to better understand the involvement of these genes in phosphate metabolism. In going down in the table, we also observed several genes related to the metal metabolism, most of them related to iron metabolism, and it was already expected because phosphate in soil has some, it competes with other nutrients, mainly iron, and these genes have a kind of antagonist regulations, the ones with related to phosphate metabolism and the ones related to iron metabolism. So we also explored this in our review. Actually, we explore each of these genes in the table in a very detailed way in our review. So going to the second group, we can see that also 35 genes were found as unknown genes with unknown function in phosphate metabolism. And interestingly, only 5 of them are genes, well-known genes, but with no relation, or at least no non-relation in the phosphate metabolism. For example, here you can see the alternative oxidase, AOX, the subunit 1E. This gene, this protein, it is very known in the respiratory chain, mitochondrial respiratory chain. However, there is no information about the relation between alternative oxidase and phosphate starvation. So the same, of course, for the other genes highlighted here. And for sure, the involvement in phosphate starvation are necessary to better understand this nutritional stress. Going a little bit down, actually having an overview of the table, the other 30 genes in the table are genes with unknown function, or they have only hypothetical function or a hypothetical responsiveness in responsive. They are responsive in some specific treatments, or they have some relation with unknown proteins, or some of them doesn't even have a name or symbol. So these ones should be considered a focus for phosphate starvation if ones want to find new regulations for this stress, or new regulations or new functions in this pathway. But in our review, then as you will see here, we also explored these genes in other crops. I mean, those were genes in rice, but we wanted to prospect these genes, which are new players on phosphate starvation response, in other crops. So we performed some autologous gene mappings based on the information retrieved from the ensemble database using the GORT2 in the gProfiler web server. So these two really helped us to quickly and accurately find the orthologs, the corresponding orthologs in maize, soybean, and wheat. And we also include the Rhabdopsis thaliana because, as I said, it's the model plant. And then we were able to generate two big tables here. One in the left for the first table I had shown here, the table with the genes with non-function in phosphate metabolism. And at the right side, we show this table with the orthologs of rice genes with unknown function in phosphate starvation metabolism. So these are all phosphate starvation responsive genes with unknown function in rice, but which can also be investigated in other crops, in other species. And this makes the thing with phosphate studies in other crops even broader. And we hope with these results can help people studying the phosphate metabolism in these species. So our conclusions and perspectives in the context of plant science are that, first, although phenotypic response to phosphate starvation are well characterized in the roots, several gaps of knowledge still exist. So here, for example, we have shown several genes which are regulated and strongly regulated by phosphate starvation, but their function in this metabolism is not known. Second, several well-known genes have unclear function in phosphate metabolism, even evidently being responsive to phosphate starvation and having functions related to this condition. So this is related to the first talk. And also 35 phosphate responsive genes never previously explored in the context of phosphate metabolism were revealed here in our results. And our analysis offered hopefully new alternatives for the development of crops with improved phosphate acquisition and use efficiency. hopefully helping to cope with the global food crisis combined with the panorama of depleting phosphorus results. Well, in the context of bioinformatics, our conclusion is that meta-analysis and systematic reviews with the use of bioinformatic tools are strong approach to integrate studies, find new gaps of knowledge, and answer scientific questions. So personally, I have the feeling that these tools should be used more often by plant physiologists, not only plant researchers actually. There are plenty of fields that these bioinformatic tools can be used, but still they are not used as it could be used. I mean, they have such a potential to answer scientific questions and to show or to reveal new scientific questions, and still they are not extensively used, at least yet. So, for example, in medical science, it's used a lot, but still there is room to improvement in biological sciences, and of course, in my research area as a plant scientist as well. So I would like to thank you for your attention. I know this presentation, our study is not a deep study in bioinformatics, but I wanted to show you the use of bioinformatics in our research as plant scientists. I feel that I had the need to study more about this. I really like bioinformatics, and I started to study even more. And hopefully, in the next event, I would be able to bring you some more something more related to bioinformatics. But anyway, I think it was important to show you the answers we could reach using bioinformatic tools, even considering that they are quite simple. I would also to say thanks to the Federal University of Rio Grande do Sul, to the programs involved, and also to the funding agencies, CAPS, CNPq, and FARPEX. I leave here my email if you have questions or suggestions or want to discuss about this presentation and the results. Feel free to send me an email, and I hope to see you next year.